Hello everyone. In today's session, we want to talk about the structure and function of the liver. We want to look at how the liver, being the largest gland in the body. The liver is the largest gland in the body. It is the largest gland. And being the largest gland in the body, it weighs 1.5 kilograms. And this liver, which is the largest, it is found in the upper hypochondriac region. That is the right hypochondriac region. And it is always inferior to the diaphragm. So we see it down, up, it is surrounded by the diaphragm. This is the diaphragm. So it is inferior to the diaphragm. And this diaphragm is the one that is protecting it, found in the right hypochondriac region, right upper hypochondriac region of the abdominal region or abdominal cavity. So that is where the liver is found. And being the largest, we see it playing very important roles. And this liver, it is divided into two lobes, whereby we have the left lobe. This is the left lobe and the right lobe. We have the left lobe and the right lobe, whereby these two lobes, the left is the smallest, is the smaller lobe, whereas the right is the larger, or it is the largest. And these two lobes of the liver, anatomically they are divided by the fusiform ligament. Fusiform ligament divides these two lobes apart. And these two lobes, each lobe, they have what we call the lobules. And these lobules are made up of millions and millions of functional units known as hepatocytes. So what we are saying is that this liver is made up of lobules. And these lobules they are hexagonal in shape. They're hexagon in shape, or you can say they are honeycomb-like. You can say they are honeycomb. And being honeycomb-like or hexagon in shape, they are made up of the functional unit of these lobules, they contain hepatocytes. So hepatocytes, they are the ones we call liver cells. And these hepatocytes forming the lobule, and they are hexagonal or honeycomb-like in shape. Those are the functional units of the liver, which is the liver hepatocytes. So this is a liver hepatocyte, and they are made up of hexagon. They are hexagonal in shape. That is what we can talk about, the functional units of the liver, which are the hepatocytes. And this hepatocyte, apart from having the functional unit called the hepatocyte, it has also what we call the biliary system. It has the biliary system, whereby the biliary system is the one in the green, whereby it is made up of the smallest portion. The smallest portion is the biliocanaliculi. And the biliocanaliculi leads to what we call small bile duct. Small bile duct. And these small bile duct are the one that give rise. This one will be the right hepatic duct. It gives rise to right hepatic duct. And this is still the biliocanaliculi giving rise to small bile ducts. And the small bile ducts, they give rise to left hepatic duct. And the right hepatic duct and the left hepatic duct meet at what we call common hepatic duct. And it is this common hepatic duct that can lead into what we call the gallbladder, this one being the gallbladder. And this gallbladder, it has what we call the cystic duct, which drain into what we call the common bile duct. And it is the common bile duct that continues into the duodenum, which is part of the small intestine. And this bile duct meets the pancreatic duct, and the pancreatic duct and the duct of Santorin at an ampulla known as ampulla of Vata. So they meet at an ampulla of Vata, or what you can call hepatopancreatal ampulla. Hepatopancreatal ampulla. So this is what is happening. So this is the biliary system. So the biliary system deals with the bile canalituli, giving rise to small bile duct, which gives rise either to left hepatic duct and right hepatic duct. And these ducts come up into the common hepatic duct, which even the gallbladder joins via cystic duct to form what we call common bile duct. And this common bile duct, we have seen it meets the pancreas, the ducts of the pancreas at the ampulla of Vata, or what we have talked about, hepatopancreatal ampulla. And this is what we are talking about. And these ducts, they are controlled by a sphincter muscle. We see them being controlled by a sphincter muscle at this point, and this sphincter muscle is called the sphincter of Oddi. Sphincter of Oddi is the one that controls the secretion of both the ducts from the pancreas and the common bile duct, where they meet at the ampulla of Vata. So this is basically the bile duct. It is the biliary system. This is what we call the biliary system, containing both of these ducts, which we have seen. So after seeing the biliary system of the liver, and this, the major function of the biliary system is the passage of the bile. The passage of bilirubin. We know that in the liver hepatocytes, we form bilirubin, and this bilirubin is conjugated within the hepatocytes, and other substances, then they drain via the bile duct, via the bile canalituli, and they are joined into the small bile duct, then into the common hepatic duct. And this bilirubin, which is direct, is always taken to the gallbladder to be concentrated, and after being concentrated, passes via the cystic duct to the common bile duct, where it is excreted into the duodenum. So we see the passage of bile being the biliary system. And in the coming videos, we shall also see, in case there is obstruction of the biliary system, which conditions shall we see. And as we conclude on the structure of the liver, as we have seen this being the portal triad is made up of the branch of hepatic artery. It has the branch of hepatic artery. It has the branch of hepatic vein, hepatic portal vein, which is this one brings nutrients from the ileum to the liver. This one is carrying nutrients, and this one is carrying oxygen. It is supplying oxygen to the liver. Then this is the tributary of the bile duct. This is the tributary of the bile duct. This is tributary of the bile. So it is from this point at the tributary where this joins the bile canalituli into the small bile duct, then the right and the left hepatic duct, then the common, then to the gallbladder. So this is the one that is responsible for carrying of bilirubin. So here the bile, or what we call the bile pigments, are transported into the, starting from the bile duct, the tributary of the bile duct. And we see from this, the tributary, this hepatic artery supplies from the hepatic artery to bring oxygen, it takes into the central vein. So we are going to see that if we have the hepatic artery brings this oxygenated blood into the central vein, and this is the central vein we are seeing, the central vein. Each hepatocyte has the central vein. And this central vein is the one that opens into what we call the sinusoid capillaries. Sinusoid capillaries. These sinusoid capillaries are the one that supplies this blood with oxygen and nutrients between the hepatic plates. Because when you have different hepatocytes, two columns of hepatocyte form what we call the hepatic plate, and this hepatic plate is whereby we see the bile canaliculi draining the secretions to join the small duct, bile duct. Then the vein, the capillaries that is separating the two plates, hepatic plates, is what we are calling the sinusoid capillary. And this sinusoid capillary is a continuation of the hepatic artery and hepatic vein, hepatic portal vein. And we said this one is bringing oxygen, this one is bringing nutrients. This one is bringing nutrients. And within the linings, this sinusoid capillaries in the epithelium, in their epithelium, we find there macrophages, and these macrophages are called the Kupffer cells. They are called the Kupffer cells. These are the macrophages found in the liver called the Kupffer cells. And these macrophages are the ones that are doing the work of phagocytosis. In the case of any worn out cell, in the case of any invasion of a microorganism, a pathogen, they are protected by the Kupffer cells found in the epithelial lining of the sinusoid capillaries. And this sinusoid capillary is the one that continues to supply the central vein, takes the blood into the central vein. Actually, this hepatic duct is leading directly to the sinusoid capillary, and then the sinusoid leads to the central vein, and this central vein supplies to the hepatic vein, and this hepatic vein is the one that gives this saturation to the inferior vena cava. Inferior vena cava, and this inferior vena cava receives this blood via the right side, the right atrium of the heart. Right atrium is received in the heart via right atrium. So this is basically the blood supply of the liver. So the liver is supplied by both hepatic artery and hepatic vein, which forms a network known as the sinusoid capillaries, where we are finding the macrophages known as the Kupffer cells, providing the protection, and this blood is supplying oxygen to the hepatocytes, and at the same time the hepatic vein supplying nutrients to the hepatocytes. Then they drain into the central vein, which gives to the hepatic vein, then transports it back to the heart. So this is the bit of anatomy. We have seen the blood supply of the liver. We have seen the biliary system of the liver and where it drains from. And we have also seen the different lobes of the liver and how that they are made up of a triad. So this is what we call the portal triad. Portal triad is made up of these branches of the artery, the vein, and the tributary of the bile duct. So this is basically the anatomy of the liver, or the structure of the liver. After seeing the structure, now we need to see the function of the liver. What is the importance or the role of the liver in our body? So the functions of the liver, I want to look at the functions of the liver. And this liver plays a big role, being the largest gland in the body. It plays a big role in metabolism, in carbohydrate metabolism. It plays a big role in protein metabolism and lipid metabolism. These are some of the functions of the liver, whereby in carbohydrate metabolism, we see this liver playing a big role in glycolysis occurring in the liver. We see glycogenolysis, glycogenesis occurring in the liver. We see glycogenolysis also occurring in the liver. So it can increase or decrease sugar levels. That is the role of the liver. That is carbohydrate metabolism. And another thing we see under protein metabolism, we see that these ammonium which are formed and very toxic in the body, they are detoxified via that the proteins undergo deamination. They undergo deamination. They are demyelinated to give rise to ammonium ions, and these ammonium ions are toxic. So they are rendered harmless by being converted to urea by the process known as urea cycle, so that it is easily or safely excreted. Then we see also lipid metabolism, whereby we see lipogenesis. In case the body needs triglycerides, they are synthesized in the liver. We see lipolysis, lipogenesis. Then we see also cholesterol being secreted from the liver. That is roles of the liver. Another thing we see playing it, this liver plays a synthetic function, whereby it synthesizes synthesis of bile salts, bile containing bile salts, and bile acid. So it synthesizes, and after synthesizing, it excretes. So it does both a lot of synthesis of bile and excretion, as we saw what was happening in the biliary system of the liver. Then we saw the liver doing the role of synthesis of plasma proteins, and these plasma proteins include albumin, fibrinogen, we have globulins. These are synthesized by the liver, and these ones form part of the clotting factors. So we see also synthesis of clotting factors. Apart from synthesis of plasma proteins, this liver also plays a big role in detoxification of drugs or toxins, and sometimes hormones. So we see drugs or toxic substances and hormones being detoxified by the liver. It does a big role there. This liver, we have seen, it has Kupffer cells which are responsible for phagocytosis of pathogens and removal of worn out cells. Removal of worn out cells. Then apart from phagocytosis by the Kupffer cells, we see this liver also doing an important role. We have seen the metabolism synthesis. It does excretion of, it plays a big role of excretion of cholesterol. And this cholesterol is excreted in the form of bile, together with the bile. We see this liver also playing a big role in digestion, whereby these bile salts that are secreted they enable the bile salts which are secreted. They enable emulsification of fats. They do a lot of emulsification or breakdown of big fat globules into smaller globules. Emulsification is when I have a big fat globule, it is broken down into smaller lipid droplets called micelles by the bile salts and the bile acids in the liver. And this process is what we are calling emulsification. Apart from excreting bile, we see also it plays a very big function in excretion of waste products of metabolism, whereby these waste products of metabolism are excreted from the liver via bile, and it is excreted in feces, whereby they are in form of stachobilinogen. So these are some of the functions of the liver we can look at in the body. And the last functions we can see for the liver, is activation. We see them, they play a big role of storage, whereby they store glucose in the form of glycogen. They do a lot of storage of fat-soluble vitamins like vitamin A, D, E, and K. They are stored in the liver. And another function is that this liver, they participate in activation of vitamin D. So vitamin D is activated in the liver. And finally, this liver, we see them carrying a big role of extramedullary hematopoiesis. They do what we call extramedullary hematopoiesis, whereby this liver in the fetus, when the embryo is still in the stomach, we see it carrying out hematopoiesis or formation of blood. Hematopoiesis is the formation of blood cells. And these blood cells in the fetus, they occur in the liver. This basically from 3rd month to the 7th month, we see hematopoiesis occurring in the liver. And also even in adults, whenever there is bone marrow failure, then the liver can come in to do a role of producing blood cells. So we have seen different functions of the liver, whereby you have seen that is metabolism of carbohydrate, metabolism, second being metabolism of proteins, metabolism of lipids, then synthesis of bile. We have seen synthesis of plasma proteins and clotting factors. We have seen detoxification. We have seen playing a defensive role of phagocytosis by the Kupffer cell and even removal of worn-out cells. We have seen excretion of cholesterol from the liver. We have seen it playing a big role in digestion. And we have seen also playing a role of storage, whereby it stores glucose in the form of glycogen. It can do storage of vitamin. We have seen activation of vitamin D and playing a big role in the production of blood cells by the process known as hematopoiesis. So these are the major functions of the liver we can talk about in this session. And thank you so much for listening from the beginning until the end. Please always remember to subscribe. Remember to subscribe, like the video, and share your comments. And we are always there for you to serve you. And this one, thank you until we meet.